Uh, Hello everyone, my name is Qada Yahya Zakaria from Algeria and I am presenting with my teammate Asito from Burkina Faso. Our topic today is should CRISPR or gene editing technologies be used to modify insect vectors to control neglected tropical diseases? We are the negative team, meaning we argue against the use of this technology. First of all, what is CRISPR? Well, CRISPR stands for clustered, regularly interspaced, short palindromic repeats. It is a gene editing technology that allows scientists to modify DNA precisely, quickly, and expensively. It was first discovered in E. coli by Ishino and colleagues in 1987. Although CRISPR offers incredible potential in science and medicine, but applying it to modify insects vectors like mosquitoes or snails raises several ethical, ecological, and social concerns. These include like risks to ecosystems, uncontrolled spread of modified genes, lack of local consent, and weak governance frameworks. Diseases like dengue are a growing health problem in many parts of Africa, mainly spread by Aedes aegypti mosquitoes, a specific type of this species. Scientists have suggested using CRISPR-Cas9 to genetically modify these mosquitoes to stop disease transmission. However, this approach raises serious concerns as the negative team. We firmly believe that disabling or limiting this technology is a better choice to protect both Africa's nature and its people. From an ecological perspective, releasing genetically modified species could lead to irreversible consequences. So, according to Collins in 2018, altering the genes of insect population unintentionally modify their behavior or causes mutations we cannot predict. Technically, Rodriguez in 2016 and Handelby and Harwood in 2019 found that CRISPR-Cas9 may create off-target mutations, unexpected DNA changes that are passed to future generations. This means once the gene drive starts spreading, we may lose control over its long-term effects on ecosystems. And beyond the science, there are also social and ethical challenges. In many African countries, biosafety policies and regulations for gene editing are still weak or unclear. So, communities often lack full information or meaningful participation in the decision making about these experiments. People have the right to be informed and to give consent, especially when the potential ecological impacts could affect their environment and health. A good example is Burkina Faso case in 2018. The National Biosafety Agency approved the release of up to 10,000 genetically modified Anopheles mosquitoes as a part of the Target Malaria Project. But while this project was conducted under scientific supervision, it sparked a public concern about transparency, trust, and long-term consequences, as Basil and Ganel in 2019 pointed out that communities often felt that their voices were not fully heard. This shows the social trust and governance are as important as scientific progress and shows there is a lack of communication between African people and these advances in genetic technologies. So I will leave this part for my fellow Asito. You can start to present.
Okay, thank you. For the negative position with CRISPR, with target malaria case, we have a ecological uncertainty. What is means? Ecological uncertainty means we don't fully know what will happen to nature if we change or remove a species. Nature is very complex, and a small change can have big, unexpected effect on the environment. For example, if we release a gene edit mosquitoes, we don't know how it will affect birds, birds, and fish that eat mosquitoes, other insects or plants that depend on mosquitoes, the balance of the local ecosystem. Irreversible risk. Once gene-edit mosquitoes are realized, the change cannot be taken back. If something goes wrong, it may take years of decades to notice, and by then it may be impossible to fix. What is matter for Burkina Faso? Burkina Faso has many small ecosystems that are important for farming and water system. Mistake could hurt local wildlife, crops, or water quality, which affect people' life. This make realizing gene-edit mosquito very risky, hard to control. Gene drive can spread quickly, but the target malaria term in Burkina Faso is moving very furiously. They have not yet released any gene-drive mosquitoes, only sterile, no gene-edited male in small, controlled field. This shows that the project is under strong control with constant monitoring and government override before any release. Community concern and justice. Many people living here near the place where gene-edit mosquito meat be released don't fully understand what's happening. Building streets, projects like Target Malaria must work hard to build trust with local community. This includes good communication, listening carefully, and sharing the benefits fairly. Lack of policy and regulation. What is the problem? Without strong rules, project can happen too fast or without enough safety check. Company or researcher will make decision without enough oversight or public input. What should be done? Burkina Faso and other countries need to create strong policies and laws about gene editing. These laws should require clear safety testing, public involvement, and government control. International groups should help countries share information and set fair rules. For the conclusion, closing statement, will CRISPR may offer solution to disease. Use it on insect vector comes with serious risks such as unpredictable effects on nature, irreversible change, a lack of clear regulation, and uncertain consent from local community. Before moving forward, we should take the time to strengthen research, build strong safeguard, and ensure decisions are made responsibly.
So
Next.
thank you, Philo Asito, for this part of Rebuttal. And we firmly welcome any questions, and we are ready to answer with what we have. And this part for bibliography resources, these are all the resources that we used to conduct this little research. We encourage people to look into these resources to have more information and have a deep sight. And thank you all for listening. And thank you, fellow Asito, for the presentation also.
Thank you for you, Zakaria.